So now we'll begin to finish up chapter 10 on gene expression. And so the final step of gene expression is translation, going from the mRNA, which we have already made and processed. It's now made its way out into the cytoplasm, and so we need to figure out how to get that from mRNA into protein. So we need to make that polypeptide chain. So we'll begin this discussion talking a little bit about the importance of the genetic code, the fact that it's universal, and then we will walk our way through the stages of translation, which include initiation, elongation, and termination. In this process of translation, we are going from mRNA into proteins by forming a polypeptide chain. So again, this is linking together a bunch of amino acids to form a particular protein. To do that, we're going to utilize the genetic code, and what this is basically a code in which particular nucleotides encode for a particular amino acid. So again, we're translating from the language of nucleotides into the language of amino acids, and so we are going to be able to assign a particular codon, which is 3 nucleotides, are going to encode for a particular amino acid. Just to reiterate, we have already made our transcript. If we're looking in prokaryotes, the process of transcription and translation can actually happen in concert together. As soon as you've got some of that transcribed product, you begin to translate it. However, for eukaryotic mRNA that actually has been processed, we've added a 5' cap. At the 3' end, we've added those adenines, our poly A tail, and the transcript has also undergone splicing. So depending upon what we're looking at, either prokaryotic or eukaryotic mRNA, we are going to have slightly different modifications to that. So when we're looking at the mRNA, we have what we call the 5' untranslated region, which means it's transcribed, it got made into RNA, but it's not where it's not actually encoding for proteins. So we know where to begin to translate that into amino acids by looking for our start codon. And our start codon is going to be a methionine. I'll show you what that looks like here in a second. So again, if we're looking at eukaryotic mRNA, it's going to have the 5' cap. Just downstream of that is going to be our untranslated region, our 5' UTR, and then we know to begin to make our amino acids once we have located our start codon, which is going to encode for the amino acid methionine. We're then going to have a series of amino acids that are going to get linked together based upon whatever is the particular mRNA, and then we know where to stop this whole process by once we come into contact with a particular stop codon, and I'll explain what those are. And then just downstream of that is going to be what we call the 3' untranslated region. So this is the region that again has been transcribed, but it's not going to be made into any polypeptide chain. And then at the end, we're going to have that poly A tail. Again, that's a form of protection from different nucleases that we might find in the cytosol. So as the ribosomes assemble onto our mRNA transcript, usually if it's a eukaryotic gene, it's going to assemble here at the 5' cap. There's a different sequence, which I'll explain further if it's a prokaryote. And it's going to scan until it hits that start codon, and then once it hits the start codon, translation will begin, and the ribosomes will just move their way down the mRNA until they hit that stop codon. So when we talk about triplet of codons, what that means is that every 3 nucleotides on the mRNA encodes for a particular amino acid. So for instance, our start codon is always going to be an AUG, and AUG is always encodes for a methionine. The way I've remembered this since I personally was in high school is that the school year always starts in August, so it always begins with an AUG. And my students have remembered that for years as well. So that's an easy way to remember what is your start codon. So again, once the ribosomes assemble there, they will move their way down until they hit the AUG, and that's where translation will begin. And so most proteins, there's a few exceptions, but most proteins do begin with the methionine, and then every 3 nucleotides will encode for a new amino acid. So again, the first one is an AUG, which is a methionine, but then we can look at the genetic code for UAC and figure out what is that next amino acid. So if we wanted to figure that out, we would look again at the mRNA. U, A is our second base, and C, that means the second one is going to be a tyrosine. And so you can move your way along again until you hit a stop codon. And as opposed to having a single start codon, which is methionine, there's 3 different potential stop codons. The way I've remembered this also since high school is, I'm sure there's a better way to do this, but I memorized it as you are good for the UAG, you are all right. I don't know why I picked that, but it stuck with me, and you go away. So anyway, usually you will be provided with a genetic code, but that has stayed with me. I know my stop codons by you are good, you're all right, or you go away. So as we previously discussed, that first AUG is methionine, and as we looked at the genetic code, we determined that UAC encoded for a tyrosine, and you can continue to march your way down the mRNA, looking at a triplet of codons all relative to that first AUG. And you will do that until you eventually hit a stop codon, either you are all right, you go away, or you are good. And then we know that we stop, and then the ribosomes will disassemble, the mRNA will disassemble, and we now will have our polypeptide chain. So that triplet of codons is what we call our reading frame. One of the really interesting things about the genetic code is that this is universal for all organisms. So interestingly, you could take a human genetic code, a human DNA, and introduce that into bacteria, and they would actually make the same protein for us, which is actually how we make recombinant human insulin. It was actually the first recombinant protein that was made, and so we can use bacteria as little factories to make human proteins for us because indeed the code is universal. So now I'd like you to look at the following problem, put the video on pause and answer the question, and then I will guide you through it together. The question asks, how many amino acids comprise this polypeptide? And you are then given a nucleotide sequence or mRNA sequence. So the correct answer here is E, which is 3. So the way you would solve this is you go ahead and you look at the mRNA sequence and you scan it until you find an AUG. And so we see one here. So we know that AUG would encode for methionine. And then next we see CCA. And again, you could use the code to figure out what that is. CCA is a proline. So we have methionine and then a proline. And then next we would have AUU, and you could go ahead and encode for that. And then next we see UGA, and if you remember, the way I taught you to remember these was you go away, which is one of our key stop codons. And stop codons do not encode for an amino acid, so this amino acid would only be 3, or this polypeptide would only be 3 amino acids in length. All right, so how is it that the mRNA is actually going to encode for a particular amino acid? Well, that is all mediated through tRNAs, also known as transfer RNAs. So tRNAs are indeed RNA, so they are single-stranded, but they have a very interesting structure because of complementary base pairing that takes place within these different portions of the molecule itself. And what we note is there's a couple of key regions in this structure. The first is the anticodon, and that is shown here. You can think of this as potentially the foot of the molecule, but the anticodon is going to be complementary to the codon that's on the mRNA. And so here you can see the mRNA has a sequence of GCC, and the anticodon is complementary to that. So the G is going to interact with the C, this C is going to interact with the G, etc. And now we know that GCC, if we go back and look at that at our genetic code, we know that GCC is always going to encode for a particular type of amino acid that is going to be held here at the top of the molecule. So we've got this structural component to the molecule because of complementary base pairing. So even though this is single-stranded, it has a very complex structure. And then we have the amino acid that is attached here via an ester bond that is specific for this particular anticodon that will be complementary to a particular codon on the mRNA. So I'm actually not going to discuss the role of aminoacyl tRNA synthetases. It's discussed thoroughly in the book, and if you're curious, you can go ahead and look at that. It's focusing on figure 10.11, so 1011. All right, so again, if we've started with our DNA and that then encoded for a particular mRNA, and we're going to have this 5' UTR, and not shown here, our 3' UTR, that's our untranslated regions, we are going to begin this whole process of translation at a start codon, and that will proceed through until we hit a stop codon. And how is it that a start codon is going to ultimately be linked to a particular amino acid? Well, that's mediated through the tRNAs, and the tRNAs are what are going to interact with that codon via its own anticodon, and then on the other end of that tRNA molecule, it is attached to a particular amino acid. So what is it that's going to bring together the tRNAs with the mRNA? That is going to occur at the ribosomes. So the ribosomes are where the translation process is going to actually take place. So an important question is, what exactly are ribosomes? Well, they are made up of proteins as well as ribosomal RNA. So if we're talking about a bacteria, that's actually going to be made in the cytosol and then assembled in the cytosol. And once we've got those ribosomal subunits, they can begin the process of translation. Eukaryotic ribosomes are similar in terms of they have a large subunit and a small subunit, just like we see in bacteria. However, if you remember our cell structure, you'll recall that ribosomal RNA is actually made in the nucleolus. And so the protein portion of ribosomes is made in the cytosol, it translocates into the nucleolus, and there the ribosomes as we know it are assembled with the appropriate protein structures and ribosomal RNA, and then that translocates out into the cytoplasm where translation can begin. Once those two ribosomal subunits come together and interact with the mRNA, we basically have three different sites within that whole unit. The first we call the A site, which stands for the aminoacyl site, and this is where the new tRNAs that are attached to a particular amino acid will interact with the appropriate complementary codon on the mRNA. The P site is where we are forming this elongating polypeptide chain, so we call this the peptidyl site. And what's going to happen here is a new peptide bond is going to be formed between the polypeptide chain that's on the from the growing P site to that A site. Once the polypeptide chain gets transferred to the amino acid that's kind of in that first position, this tRNA will now no longer have the amino acids or polypeptide chain attached to it, and so it's going to enter into what we call the E site or the exit site, and then that empty tRNA that is no longer interacting with the amino acids will be able to leave the E site or the exit site and go and find the appropriate amino acid once again, utilizing the amino acyl tRNA synthetase, which is described in your book, and be able to repeat this whole process. So when we're talking about translation, we have three key steps. We have initiation, elongation, and termination, and I'm going to walk you through each of those. The first is shown here. This initiation is when the small ribosomal subunit and the mRNA are going to interact. We're going to get the appropriate initial tRNA that we know will be bound to a methionine, because the AUG is where this is all going to begin. And then the large ribosomal subunit will kind of simultaneously bind, and we now have our whole ribosomal subunit ready to begin the process of translation. We will then see elongation taking place, in which tRNAs with the appropriate amino acid will interact here with the appropriate codon because it's got a particular anticodon that's complementary. That peptide, that polypeptide chain will get transferred to this particular tRNA, and then the tRNA that's no longer bound to the polypeptide chain will enter into that E site and dissociate, and that's all going to progress until we ultimately hit a stop codon and the whole thing will disassemble. So I'm going to walk you through those steps in a little bit more detail. So with regards to initiation, we have three key things that are going to happen. First of all, the mRNA, that first tRNA that's bound to the methionine, and the ribosomal subunits need to assemble. This is going to require the assistance of some ribosomal initiation factors, and this also takes energy. So the energy in this particular case is going to come from GTP. So when we're talking about prokaryotes, the way this will begin is not at a 5' cap, but we're going to have a particular ribosome binding site. And as I said, whether we're talking about prokaryotes or eukaryotes, the initiation of translation is going to begin at an AUG. So we see the small ribosomal subunit is going to interact with the mRNA, and it's because we've got this ribosomal binding site. So it's a sequence of mRNA that encapsulates that AUG, and the ribosome, that smaller ribosomal subunit is, that's an indication of where it should be attaching to the mRNA. We then will get recruitment of that large ribosomal subunit, and then this whole ribosome will make its way down towards that 3' end of the molecule. All right, so again, what we see here is that small ribosomal subunit, and there's going to be particular sequences in the mRNA that will indicate that this is where indeed that small ribosomal subunit should bind. We see encapsulated within that is going to be our AUG, our start codon, and so that initiator tRNA that's bound to a methionine is going to form the complement. So here we have our AUG, the complement is UAC, and that is going to be, that tRNA is going to be attached to a particular ribosome, and then we'll get recruitment of that large ribosomal subunit, and then the whole thing will begin to make its way down towards the 3' end of the mRNA chain. Now, translation is slightly different in prokaryotes versus eukaryotes with regards to this initiation step. So whereas in prokaryotes they have that ribosomal binding sequence, that is not true for eukaryotes. We know that eukaryotes get that 5' cap, and that's actually one of the key indicators for where the ribosome should be attaching to and assembling, which is great because that's at the very beginning of our mRNA sequence, and then it's going to make its way down until it comes into contact with an AUG. And the AUG, it might not necessarily be the very first AUG that it comes into contact with. This is showing the DNA sequence, which would be ATG. But oftentimes it is. We will find something called a Kozak sequence, which is, it's not just the AUG, but it's also some key nucleotides that we find just adjacent to the AUG. And so this is where the ribosomes know to initiate translation. So oftentimes we'll have that AUG, but usually just downstream of that AUG, oftentimes is going to be a G. Just upstream of that, most of the time you're going to see either a C or a G, and we call this the Kozak sequence, named after the woman who discovered it. Okay, so now we're going to move on to elongation. So that was initiation. Elongation just means we're going to now attach on and have our growing polypeptide chain. So once the AUG is bound in that very first position, which is really our P site, that means the A site is now established and ready to welcome in a aminoacyl tRNA with an appropriate amino acid. So once that happens, then a peptide bond is going to be formed between the P site and the A site, so that amino acid that was in the P site, which in our case for the very first amino acid is going to be a methionine, that's going to get transferred to the amino acid that's currently in the A site, and now we're going to have our growing polypeptide chain. And so those ribosomes are going to move down and translocate towards a 3' end. And that's shown here. So this is an already established polypeptide chain. Here we call, again, this is our P site. The new liberated free A site is ready to welcome an appropriate tRNA. So here we are going to form the complement to CGU, which is going to be GCA, and that is bound to a particular amino acid. And then what we're going to see is that polypeptide chain in the P site is going to get transferred to the A site. And then those ribosomes are going to move down. So effectively, once that happens, what had been in the A site is now going to become the P site. What had been in the P site is now going to enter into the E site, the exit site. And that tRNA that is now no longer bound to any amino acids, or that polypeptide chain, is going to exit and go reattach to a new amino acid. And now we have our polypeptide chain attached to the tRNA in the P site, and now we can welcome in a new tRNA to the A site. And so that's elongation. And that's going to just progress through until ultimately we hit one of those stop codons that we've already discussed. And this whole process takes energy. Again, that's going to be in the form of GTP. Now, one thing that's really important to point out is that we will oftentimes refer to a protein as having an amino terminus or a C terminus, the carboxylic acid terminus. And so what that means is the very first amino acid, the methionine, that's going to be our 5', or that's going to be our amino terminal end, whereas the other end of our polypeptide chain is going to have this carboxylic acid on the end. So we call that our C terminus. And so that's a really important way for you to be able to differentiate where this all began and where it is ending. Okay, so again, that growing polypeptide chain, here we've got all of our amino acids. This would be our P site. This is our free A site. And we are going to make our way down, forming that new polypeptide chain, which is going to be formed here. And now we've got an empty E site. This is our P site and our A site. And once that new polypeptide chain gets formed, what had been the tRNA in the P site is going to move to the E site, and this will go ahead and exit from the ribosomes, and the ribosomes will translocate and make their way down this chain. All right, so the final step of this whole process is something we call termination. And what's going to happen in termination is we are going to come into contact with one of those stop codons, and then everything is going to disassemble. So let me show you what that actually looks like. So again, we are looking for one of our stop codon sequences, UAA, UGA, or UAG. And once we come into contact with those, we know it's time to stop translation. Once that happens, so here we've got our polypeptide chain attached to a tRNA, but once we've hit one of our stop codons, in this case it's the UAA, that is going to interact with something called the release factor. And the release factor is going to be important for a series of events that need to occur. So first of all, what happens is we see that the tRNA is dissociated from the polypeptide chain. That kind of gets hydrolyzed and liberated, and now this protein is able to go ahead and assume its appropriate folding. And then the ribosomal subunits will dissociate, that release factor will also dissociate, and the mRNA will dissociate as well. Now, one final note is it's really interesting to recognize that this process takes place extremely fast. You can make a protein in half a minute to a minute. It's all going to depend upon the size of the proteins, but it's incredibly efficient and fast. So I will end here. What I'd like for you to do is go ahead and pause this part of the video and try and walk your way through initiation, elongation, and termination. And at this point, you should be able to describe that to a colleague.